Hello and welcome to part 3 of this lecture series on the different experiments that we were talking about in lab periods 3 and 4 and weeks 3 and 4 of our remote course. I just wanted to take this short video to cover DNA transformation and some electrophoresis. These are two techniques you probably have learned about in lecture, and we do use them in the laboratory just to introduce some concepts in biotechnology and how microorganisms are an integral part of the biotechnology field. Now, the first experiment in DNA transformation, here we are going to use bacteria to actually produce some type of protein that is from another species. In the very first biotechnology related, what we call recombinant DNA product ever put on the market was insulin. This was created back in the 80s, and diabetes was a really big issue. And getting insulin that did not promote resistance, that people were able to take that insulin and not have resistance develop to it, or that insulin to be most effective, they really needed human-specific insulin. Insulin produced by other species is not quite as specific for our cells as, of course, our own. Researchers found that they could take the gene, the human gene that is responsible for production of insulin, take that gene and insert it into E. coli bacteria. They added the gene to a plasmid and then inserted that plasmid into bacteria in order to have E. coli bacteria produce insulin, and the insulin they produced was human specific. To this day, we still use genetically modified E. coli to produce human-specific insulin for diabetics. And this changed the treatment for diabetics over time drastically. And so diabetes used to be a fairly deadly disease, and people would die from it at a much younger age, and now people survive well into old age with diabetes due to this introduction of human-specific insulin being produced by bacteria. This same concept is now being used for lots of different pharmaceuticals, and it's a really kind of ingenious process. All right, so in our experiment, we're going to make bacteria that are antibiotic resistant by introducing a plasmid containing that resistance gene. We're going to introduce that gene into some E. coli. So we start with our gene. We have our gene of interest. In this case, we are using a plasmid called PUC18. PUC18 is the name of the plasmid. And on this plasmid is a gene for antibiotic resistance. Now, this bacteria that we're using in our experiment, it's E. coli and ampicillin. So the E. coli that we have in culture is susceptible to ampicillin. It is incapable of growing in the presence of ampicillin. PUC18 plasmid contains a gene on it for that antibiotic resistance, for ampicillin resistance, and it is referred to as the AMPR gene, AMPR, capital R. This is called the AMPR gene. What we're going to do is we're going to introduce this PUC18 plasmid into the bacteria cell. Now, transformation, you may have learned about, is a form of horizontal gene transfer that bacteria use to exchange genetic information. What we're doing is we're taking this natural process that bacteria would use in the environment when they come across each other and they want to exchange genetic material. This is known as transformation. We are going to just mimic that process in the laboratory setting. So we're going to induce one set of cells to take up a protein or a piece of genetic information and get that bacteria to then start actually transcribing and translating the plasmid or genetic information that we have introduced into them. So here we have our plasmid. Here's the PUC18 plasmid. It contains that AMPR gene, the ampicillin resistance gene. Here's the plasmid right here. We're going to put this plasmid into a test tube with bacteria. Now, the bacteria themselves have been exposed to calcium chloride. And this is done on ice. They're introduced to calcium chloride on ice. And this will create bacteria cells that are referred to as competent. Now, competent bacteria cells are cells that are ready to take up DNA. Normally, in a regular environment, in their own kind of natural environment, the bacteria would come across some kind of chemical signal, or the presence of genetic material would cause them to initiate this competence, this state of competence. And in the state of competence for absorption of extraneous DNA, we just introduce them to a chemical signal, calcium chloride. Now, we do this on ice because we want to inhibit any kind of cellular leakage. And so the ice, the cold temperature, helps to kind of solidify the bacteria a little bit so we don't get too much leakage of cellular content during this competence. Now, this is because we are exposing them to a fairly high level of calcium chloride, and this will take our normal bacteria cell and basically make it kind of spongy. Kind of drill, put these little holes in it here, in E. coli in its outer membrane, and make this bacteria a little spongy and capable of drawing in the plasmid itself. So we mix this calcium chloride bacteria with our plasmid containing our gene of interest. We put them together in this test tube here, allow them to sit on ice for about 15-20 minutes, allowing the absorption of that genetic material. Once that's occurred, we then heat shock the bacteria. We put the bacteria into a heat block. We allow it to heat up at 37 degrees Celsius. Since we're using E. coli, it grows best at that rate. And this is going to shock the bacteria into repairing their cell membrane. They'll repair the cell membrane. We then take them off of ice. We then add some nutrient media, such as some nutrient broth. Then we put them back in the heat block at 37 degrees for about 20 to 30 minutes. This allows the bacteria to begin growing and to begin transcribing and translating the gene that we just introduced in the first step. So we introduce the gene, we allow the bacteria to heal, we then add nutrient broth, we put the bacteria back in its optimum growth environment, and we allow the bacteria to begin the transcription and translation process of producing our gene of interest. Once we've done that, we're then going to plate our bacteria. The experiment that you see in your lab manual, we actually plate the bacteria on an agar, it's nutrient agar, that has been subsidized with some ampicillin. So ampicillin has been added to this media. Because ampicillin has been added to the media, organisms or bacteria that did not take up our gene of interest will not grow. And the bacteria that did take up our gene of interest will be able to grow. So when we plate this, we have on one side, we have non-transformed bacteria. So this is bacteria that went through all of the same transformation steps, just with water instead of a plasmid, kind of like a negative control. Then we have our transformed bacteria. And so these are bacteria that went through the transformation process, but they did so with a plasmid, not with water. So it would be like a positive control. So here on the left, this is our amp agar. Here on the left, we're not going to have, so this is our negative. We're just going to say negative, and this is our positive. On the negative side, we're not going to have any growth. We have the Petri plate. It has our agar in it. We draw a line down the back of the plate so that we have our positive on one side, our negative on the other. And in our negative side, we're not going to see any bacterial growth. Why? Those bacteria did not gain access to this plasmid right here that contained our resistance gene. So these bacteria are still susceptible to the ampicillin, to the antibiotic. Here on the right, however, we start seeing small little areas of growth, small colonies of growth, and these are the bacteria that did take up our plasmid. So that test tube was exposed to the ampicillin, the AMPR gene on the PUC18 plasmid. Some of the bacteria that were in that culture successfully incorporated that gene and successfully transcribed and translated that gene so that those bacteria are capable of becoming ampicillin resistant. So we would see growth on the positive side of the plate and no growth on the negative side of the plate. Now we take a look at our DNA electrophoresis. In DNA electrophoresis, we were not able to do this class activity because this website is no longer working. But here we look at the use of a DNA fingerprint to determine the source of genetic material. In order to carry out this particular process in our lab, we are using what's called RFLP analysis. And in RFLP analysis, this stands for restriction, fragment, length, polymorphism. I know it sounds like a big long term here, but it's just restriction, fragment, length, polymorphism, or RFLP analysis. And what we are looking at is DNA being cut. Now, restriction enzymes are enzymes that are normally produced by bacteria, and these enzymes are used by bacteria to protect them from bacteriophages, which are bacteriophages are viruses that infect bacteria. And these restriction enzymes will cut genetic material, DNA in particular, will cut DNA in very, very specific sequences known as palindromic sequences. And they they look for specific sequences in the DNA, and they'll cut in between two bases that are within that sequence. And what this does is this causes the DNA to now kind of split apart. The DNA will come apart. Now, from one individual to the next, how often those specific sequences occur and where within the genome they occur are going to be different. And this allows us to cut different DNA, different sources of DNA, into a multitude of different fragments. And this can provide us with a kind of what we call a DNA fingerprint. We then use the process of electrophoresis to separate our DNA. So electrophoresis is a tool used to separate DNA fragments by size and by charge. Here's how it works. Over here we have a gel, right? This is an agarose gel. And at the top of our gel we have little wells. These little wells are going to contain sources of DNA. Now I have 1, 2, 3, 4 tubes here, right? So we'll just number them. Now, in my first tube here, I have a known source of DNA. And in my other three, I have three sources of DNA that I'm not sure. These are my... So here I have 4 tubes. The first 3 are from known sources of DNA. I know where these have come from. In my 4th, this is unknown. And what I mean by this is I know I have DNA there, but I don't know what the source of that DNA is. Over here, these first 3, tubes 1, 2, and 3, I extracted that DNA from a known source. So I know exactly where that DNA came from. What I don't know is I have DNA in my unknown tube, but I don't know where that DNA came from. Maybe that's DNA from a blood sample at a crime scene. Maybe that's DNA from a bacteria that I don't know the name of that bacteria. Maybe that's DNA from a virus that is causing disease in a patient, but I don't necessarily know what that virus is. I'm going to use what's known as the genetic fingerprint, or the DNA fingerprint, of my unknown DNA source and compare it to my known sources to see if I can't figure out where it came from. This works especially well with bacteria because bacteria reproduce by binary fission. So their DNA, they completely replicate their DNA. It's cloned. So now I have my 4 different samples here. I have my electrophoresis gel here in the right, and I have the 4 wells for each of my DNA samples. This gel is going to be placed inside of an electrophoresis chamber, and inside that chamber it's going to be covered with buffer. We have a buffer environment, a liquid environment, and this chamber allows us to apply an electrical charge. This side, nearest my wells, is the negative charge, and the far end of the gel on the other side of the chamber is the positive charge. And my electrical current is going to go from negative towards positive. The reason we do this is because DNA is negatively charged. So DNA that's in these wells here, when I apply this current, is going to automatically be attracted to this positive charge. And since my positive charge is the same all the way across my gel, this is going to have the same pull of those DNA fragments through the gel. Now, prior to uploading my DNA samples into the gel, I have exposed all 4 of the DNA samples to a series of restriction enzymes in order to cut the DNA into fragments. If tube 1 is the source of DNA that I have in tube 4, then those number of fragments and the size of those fragments are going to be identical, hence the term DNA fingerprint. So let's just draw in some samples here. So I apply my electrical current to my electrophoresis here, and this begins to draw my DNA in large fragments across my gel. I can just draw these fragments in here. Pardon how unstraight this is. All right, so here I have tube 1, 2, 3, and my unknown here in 4. And if I compare, if I look at how my fragments have run, my fragments have been pulled down the gel from negative towards positive charge. The larger the DNA fragment, the slower it's going to move through that gel matrix. My really small DNA fragments will run very quickly. So they're going to be down here at the bottom. They're going to run very far. So these are my small ends. And my really large fragments are going to take much longer. That agarose works like a matrix. And so they have to kind of weave their way through this polymer matrix. And so those shorter fragments are going to move really quick, and the really big fragments are going to take much longer to move across. So this is how we use DNA electrophoresis to separate our DNA fragments according to size. Large run really slow, small run really fast. And by charge, my DNA is being pulled through the gel from negative towards positive. Now I can take a look and compare my lanes. I can see lane 1 has this DNA fingerprint here, lane 2, lane 3. Now I'm going to take a look at lane 4 because I want to know if lane 4, the DNA used and cut for lane 4, comes from any of the other 3 sources, source 1, source 2, or source 3. And I can see here that all 3 of these lanes have this fragment in common, but 3 does not match. So 3 is already pulled out. Then I can just look at my second fragment here. It matches number 2. Number 1 does not have it. So it looks like it's probably going to come from number 2. But we can't stop there. We have to continue, and the entire fingerprint must match in order for it to be considered the source of the DNA. So as I move along, I have these 3 bands here. Well, I have those here too. I match here and here, here and here. So it looks like all of our bands match, and since they all match, they're in the same place and the same number of fragments, the same length down the gel, I can confidently say that the source of DNA in sample 4 is the same as the source of DNA in sample 2. Well, sample 2, I know what organism I derived that from or where that particular sample came from, so I can then relate back to that. This is how electrophoresis is used to help compare organisms, find the orientation of an organism or where an organism may have come from. So let's just do a quick review for question number 1. How are DNA fragments in an agarose gel separated during electrophoresis? We know that this is by charge and length or size, so we're going to go both A and C. And in question number 2, where on an electrophoresis gel would the smallest DNA fragment be found? Remember, these guys have to move through a matrix of gel, so smaller fragments are going to move faster than larger fragments. This means we're going to find it closest to the positive cathode, to the positive end, because it's moving towards the positive charge. So those are the two biotechnology experiments that we do in our laboratory. I hope that this overview helped you. Please make sure that you read the lab manual on this, because in DNA electrophoresis, the gel, there's a few sample gels in there that you might want to look at that would be very helpful in helping you understand how we can use this particular biotechnology tool. See you in the next one.